Up till now in this class, we've considered diseases that involved autosomes. We're now going to look at diseases that potentially are on sex chromosomes. So this is referred to as sex-linked inheritance. So normally both male and female humans have 23 pairs of chromosomes. 22 pairs are called autosomes. These are the non-sex chromosomes, and then one pair is the sex chromosomes. In mammals, there are two varieties of sex chromosomes. There's the larger one, which is called the X chromosome, and the smaller one, which is called the Y chromosome. The X and Y are mostly non-homologous, and this means they contain different genes. However, there is a region at the regions at the ends of the Y chromosome and regions that are homologous with regions on the X chromosome. So there is a little bit of that, but in general, we're going to consider these to be non-homologous chromosomes, okay? Here's a karyotype of a normal female. So she has 46 chromosomes altogether. If we look at her sex chromosomes, she has 2 X chromosomes. We have a normal male. He has 46 chromosomes also. If we look at his sex chromosomes, he has an X chromosome and a Y chromosome. Note that the X chromosome is much longer. This has implications because it means that the X chromosome carries more genes than the Y chromosome. So sex-linked traits are going to be controlled by genes on the sex chromosomes. X-linked genes are found on the X chromosome. There are about 2,000 genes on the X chromosome. We have genes for many characters unrelated to sex on the X chromosome. So for example, we're going to be talking about color blindness and hemophilia. Those are located on the X chromosome. In these cases, for the color blindness and hemophilia, there's no matching gene on the Y chromosome. There are Y-linked genes that are found only on the Y chromosome. There's about 200 genes on the Y chromosome. Most of these Y-linked genes are actually related to sex only. In other words, they help to determine the sex of the individual. We're only going to consider X-linked traits because there's so few Y-linked traits that are really involve disease. So we're going to just look at X-linked traits. So if you think about an X-linked trait for a male means that the X would have had to have come, the X chromosome would have had to have come from his mother, right? So a male always receives X-linked alleles from his mother, from her X chromosome. So X-linked traits cannot be passed from father to son. And I think the reason is obvious because the father has to contribute a Y chromosome. Even though the father is both an X and a Y, if he were to contribute an X chromosome, well, and the female makes 2 types of gametes, X and, well, no, just a single type of gamete in terms of sex, which would be the X chromosome, then you don't have a male, you have a female, right? So a male can only receive X-linked alleles from his mother. Now, this is important because later on we're going to talk about diseases that are passed down from generation to generation. And you'll see that many females are carriers for some of these X-linked traits. So again, it says here the Y chromosome from the father does not carry an allele for the trait. Most X-linked genetic disorders are recessive. For a recessive X-linked trait to be expressed, the female needs 2 copies of the allele. So she would have to be homozygous. So for example, here, imagine we have a gene B. This is going to be the gene for color blindness. If she had 2 small b's or lowercase b's, then copies of that allele, then she would be homozygous and she would in fact be colorblind. If she had the other combination be X superscript big B, X superscript big B, then she wouldn't she wouldn't phenotypically show any color blindness. If she was X with superscript big B, X superscript small b, she would just simply be a carrier and she wouldn't show color blindness. The male, on the other hand, needs only 1 copy of the allele. This is referred to as hemizygous. Hemi means half. So, and the reason for this is, again, the Y, for this, let's imagine this is color blindness. There is, in fact, no gene for this on the Y chromosome. So the male has to get the X chromosome with the allele from his mother. In this case, we see X with a small b. That would mean that he would be colorblind, for example. X-linked recessive disorders are much more common in males and females. In most cases, females are carriers. And I think you can see why this would be the case. The female can only express the disease if she actually has 2 bad copies of the gene, whereas the male just needs to get 1 bad copy of the gene. And he's not protected essentially by a second copy of the gene or second allele because he has a Y chromosome, which simply does not have the gene at all. Females must receive 2 recessive alleles to have the disorder, 1 from each parent. So I've already mentioned this, but let's go ahead and look at red-green color blindness. That's going to be our example that we're going to study a little bit here. It's much more common condition in men than in women. So if you look at Caucasians in the United States, perhaps something like 1 in 12 or so men will have it. But it's relatively rare, like more on the order of, say, 1 in 200 women will have this. So color blindness is an X-linked trait. And so if we look at the genes, X superscript big B helps to lead to normal vision. X superscript small b would be a defective gene, essentially, that would lead to color blindness. So let's look at the possible genotypes. If we have X superscript big B, X superscript big B, you have a female with normal color vision, and she, in fact, does not have the defective gene. If you have a female who's X big B, X small b, she's a carrier, but she herself has normal color vision. If you have X small b, then this would be a female who's colorblind and who obviously can pass the defective gene along to the next generation. A male who has normal vision would be X big B Y. So we just a single copy of the gene, but it's the correct or normal one. However, if you have X small b Y, then this male would be colorblind. Let's look at some examples of how this X-linked recessive disorder could be passed on. And again, we're really using this. We're talking about color blindness, but it could really be any X-linked recessive disorder. What we're talking about now would hold true for any other X-linked recessive disorder. So let's take a male who shows color blindness. And here we've switched this is an illustration from a different textbook, so they're using slightly different letters. They're using small n to mean that the person is colorblind, whereas a big N means that they have normal vision. So here we have a male who's colorblind, so he's X small n Y crossed with a female who's X big N X big N. Okay, so we can imagine what kind of gametes these two would have. The male can make Y gametes, and he can also make gametes which are X small excuse me, small n. Now, the females, she can make only X big N and X big N, right? So if we look at the outcome here of this cross, then we see that the male can't pass the gene on, even though he's colorblind. To males, when he makes if he has a son, he can't possibly pass on the gene for colorblindness to his son because he has to pass the Y chromosome along. Okay, in this particular case, the female has 2 good copies of the gene, so you can see she's X big N and X big N. So the male essentially would not have in this family, they wouldn't have the gene. Okay, I'm looking for my highlighter here. Okay, so now let's go over here. Let's look at what happens in the case of females. So here, the female passes on only the big X big N gene or allele, whereas the male only passes along the X small n allele. Oh, here's what I'm looking for, right? So we have X big N, and then X small n. So all the females, all the daughters in this family would be carriers. They would be heterozygous. Let's look at another possibility. Over here, we have a female who's a carrier. So she's X big N, X small n. Now she's crossed with a male who is X big N, Y. Now let's look at what happens in terms of the offspring here. Like if we look at the Y, if the male, if we look at the sons, well, the male has to pass on the Y. And the female can pass on a good copy. So with 50% of the time, she would pass on a good copy of the gene, which would be X big N. But 50% of the time, she would pass on X small n. So there's a 50% probability, if the female is a carrier, that her son will have color blindness. Now let's look at what happens to the daughters in this family. Well, okay, here in this case, the male is unaffected. He has normal vision, so he can pass on his X big N. And the female can pass on X big N, so that's one possibility here. So this would be a female who has normal vision and who is not a carrier. Now, we have one last possibility here, which is the male passes on his X big N allele, and the female with a 50% chance passes on X small n. So this female then would be a carrier. Let's look at the last possibility over here. We have a male who's affected, he is colorblind, crossed with a female who's a carrier. She's heterozygous. Okay, so what are the possibilities? Well, the male can pass on his Y chromosome. And then with a 50% probability, the males in the family, the sons would have a chance of having being colorblind because they could get X small n from the female. And then, of course, 50% wouldn't get it. They get the X big N. All right, what are some other possibilities? Well, the male, if we look at the females, the male can pass on X small n, and the female has a 50% probability of passing on X small n. And so that would be a female who actually exhibits the disease. She's homozygous recessive, and so she would be colorblind. What are some other possibilities? Well, here's the other possibility. The male has no choice. He passes on his X small n, so that's shown here. And then the female with a 50% probability might pass on her X big N. So then this female would be a carrier. We're going to now look at a pedigree for an X-linked disorder. We're going to use the example of color blindness, but it really could be any disease. In this particular family, we have a grandfather here, and he is affected by the disease. So he has color blindness because you notice he's big X, X, small b, Y, of course, because he's male. Now he's crossed with a female who's X big B, X big B. So she does not carry the disease. Let's see what happens in the next generation. Well, he happens to pass, if he has a son, well, he's not going to pass the disease on, right? The color blindness on, because he passes his Y along, and the son gets a copy of X big B from his mother. Remember, his mother is not a carrier. She does not have the disease at all. Now, this particular male happens to get married to a woman who is X small b, X small b. So if they have a son, which is what they have here, he would necessarily 100% get the disease, the color blindness, because he passes a big Y along, and then the mother passes on X small b. So there's a 100% chance the son would get color blindness. Now, if we look over here, in this particular case, this daughter, she's actually a carrier. And this makes sense because the mother passes on X big B. The father, if he's going to have a daughter, he has no choice but to pass on X small b. So the daughter must be a carrier. Now, she marries a male who's not colorblind, so he's X big B Y. And then you can look at the various possibilities. They have a son So this is the grandson, of course, of the original generation, who was, this male passes along his Y, and with a 50% chance, the sons will have color blindness. So this one happened to get color blindness because he got X small b. Now here's a female who's a carrier. So she's X big B, X small b. Here's a female who's unaffected. She does not have the gene for color blindness. And here's a male who's not colorblind. So there's various possibilities when you have one of the parents being the female having being a carrier. As I mentioned earlier, about 1 in 12 Caucasians males have red-green color blindness. There are different forms of red-green color blindness, so different levels of severity. So this corresponds to about 7 to 10% of Caucasian males. So for them, bright greens are seen as tans and reds as reddish brown. A red-green colorblind individual shouldn't be able to read the number 74 as all the dots appear the same color. So let's take a look at another pedigree here. So we have, in this case, a male over here, and he is affected, and we have a female who's a carrier. Now, in the next generation, note that all the females are in fact carriers. And that makes sense, because if you're going to have a female and the male is affected, so he's X small b Y. So in order to have a female, he has to pass along, the male in this case has to pass along this defective gene. So all the females in the next generation would have to have it. Now, this male here in generation 1 cannot pass the defective gene, the one for color blindness, on to the next generation of males, because he has to pass his Y chromosome along. So this male here is colorblind, not because of the father, but because of the mother, because she's a carrier. Now let's look at what happens in the next generation. Here we have 2 carrier females. They cross or get married to males that are unaffected, and yet they're going to, with a certain probability, pass on the gene to the male. For example, 50%, there's a 50% probability that the male would get it. Again, in the next generation, again, the male is not getting it from his father. Well, for one thing, the father he doesn't have the gene at all, but the father has to pass on the Y chromosome. So if he gets X small b, it has to be coming from the mother. Same thing is the case over here with this male here. And for these 2 females in the next generation down, they're carriers because of their mother, not because of their father. The father doesn't have the defective gene. And then finally, we see down here that same situation. So these females marry males that are unaffected. They don't have color blindness, but a certain percentage of males have it. So should be 50%. 50% of the males will have it. And then about 50% of the females will be carriers. So that's it. So basically the general idea is that with an X-linked recessive disorder, it's much more likely that males will actually be affected. Notice in this pedigree here, there's quite a few males that are affected. There isn't a single female that's affected, although there are quite a few females that are carriers. So a good question is, well, why do we care about this? Is it really important? Is color blindness certainly isn't a very, extreme disease that really matters very much. But there are a number of diseases that are X-linked recessive disorders that are actually quite, quite significant diseases. Here's Duchenne muscular dystrophy. This is the most common type of muscular dystrophy. There's a number of different types. Duchenne muscular dystrophy is characterized by wasting away of muscles. Symptoms include a waddling gait, toe walking, and frequent falls. And we know now that it's caused by the absence of a protein called dystrophin. And so what happens is that calcium leaks into muscle cells, skeletal muscle cells, and these muscle cells then die. And the muscle cells are then replaced with fibrous tissue. So you can see that here. Over here, there's muscle that's been replaced by fibrous tissue. The fibrous tissue, of course, is not able to contract. It doesn't really help the muscle. But so what you'll see as a characteristic is these children will have very thick legs, for example. And that's because the muscles have gotten thicker, but they're actually very weak. And so again, they have frequent falls. They don't have a lot of strength, even though the legs look large. Now, this disease, because it's an X-linked recessive disorder, it affects males much more often than it does affect females. Here's another X-linked recessive disorder. It's called Fragile X syndrome. It's caused by an abnormal number of repeat sequences of cytosine guanine in the X chromosome. So we haven't really talked about this, but there are some diseases in which we have repeated sequences, typically triplets like this, over and over again. And this is actually the case in Huntington disease, which we talked about earlier. Now, it's actually normal to have these repeat sequences. In other words, you'll have CGG and then CGG again, and then again and again and again. And it's fairly normal to have, say, 6 to 50 repeats, and that's actually seen as fairly normal. There's nothing really wrong with that. If you have 50 to 200, you would be considered to be a carrier if you have 50 to 230 of these repeats. If you have more than 230 of these repeats, then you'll have the disease. So this is, in fact, Fragile X syndrome is the most common cause of inherited mental impairment. And this can range from mild learning disabilities to much more severe intellectual disabilities. It's associated with some forms of autism, and it affects males much more than females. So males have certain characteristic physical abnormalities. They'll have a long nose, ears that stick out, and use typically short stature. The name Fragile X comes from the fact that if you look here, before we really understood much about the disease, people did note in the karyotypes that the X chromosome looked fragile because there was a very, even when the chromosome was condensed, there was a very skinny part. So it looked as if it was fragile. Okay, and here we can see a lineage here where we have individuals, they're actually unaffected. So this is a female here who has less than 50 repeats. Here we have a male who appears to, who's a carrier, who actually has the disease. Actually, if he's he's a carrier from 50 to 230. He's unaffected, but he's a carrier. Now they have a daughter. Remember, circles are daughters. She's also a carrier because she has 50 to 230 repeats. Now she marries a male. Who does not have the disease at all. So he has fewer than 50 repeats. But then in the next generation, they have a number of sons, and 2 of them actually have the disease. And one appears to be a carrier because he's got 50 to 230. These 2 here have more than 230 repeats. Another very well-known X-linked recessive disorder is hemophilia. This is a disease caused by the absence or minimal presence of clotting factors. Clotting factors are plasma proteins that help in the blood clotting process. There are 2 types of hemophilia that are passed on this way. There's hemophilia A, which is caused by a lack of clotting factor 8. And there's hemophilia B, which is caused by a lack of clotting factor 9. So these people, they do have platelets. Okay, so that's one thing people often think with hemophilia. Oh, maybe these people are missing platelets. No, they do have platelets. In clot formation, let's say, imagine you have a cut in a blood vessel, the first thing that happens is a platelet plug forms, and that does involve platelets. But these platelets release signaling molecules, and this should actually cause proteins to be activated. And so that's what we're talking about here. These are plasma proteins that should naturally be present, and they're just simply missing in hemophilia A and B. So what's the outcome of this? The affected individual's blood either does not clot or clots very slowly. They can bleed excessively from either external injury, but also they can bleed from internal injury, especially at the joints. So this is a pretty terrible disorder, but nowadays those clotting factors are just simply proteins. So if a person with hemophilia cuts themselves or is injured internally, they can simply take an injection of these clotting factors, and then they would be able to clot. Their blood would clot normally. Okay, this is a family pedigree, and I know it looks very intimidating because of how large it is, but we're just going to take a quick look at it and try and understand what's going on in it. So we're going to start with Queen Victoria. Over here, you can see that she's a carrier. You'll note that neither her mother, Victoria, or Edward appeared to have the disease. Now, it's possible that it arose. So some of these diseases can arise as mutations. So it may have arisen in the germline of Victoria, or it actually may have been in the germline of either her mother, Victoria, or Edward. So we don't really know for sure. But all we know now, we know for certain that she definitely had the disease. Okay, now she married Albert, and they did have a Leopold, a hemophiliac male son. So he got the disease. It's a recessive X-linked gene. Now, if we go on, you'll see that she had some daughters. Now, some of her children didn't get the hemophilia at all, but some did. So Alice, for example, did, and then Beatrice did. And then if you look further down in the lineage here, you can see there are more female carriers, but really no females that were affected, but quite a few males that were affected. And so same thing down here. Now we're going to follow this one line over here. Now this leads all the way to Philip and Elizabeth II. Now, at some point along here, I think it's very early on, number 2 here is Edward the Seventh. Now he did not get the gene. He was not a hemophiliac male. And so from then on, you see that the disease, they're completely free of the disease. They don't have hemophilia whatsoever. And that ends with modern day, for example, Prince Harry, Prince William, they don't have the disease, so forth. Okay, that's it for this slide. So far, we've talked about X-linked recessive disorders. Those are much more common than X-linked dominant disorders, but X-linked dominant disorders do exist. One example is incontinentia pigmenti. This causes discoloration of the skin. So if you're an affected male, you can pass this trait on only to your daughter. And that makes sense because it's X-linked. It's on the X chromosome. If you have a son, you're passing on a Y chromosome to your son. However, if you do have a daughter, because it's dominant, she will have a 100% chance of inheriting the disorder. Females can pass the dominant allele to both the sons and daughters, causing the disorder. So an example of this, if the female is heterozygous and her partner is normal, then each child has a 50% chance of receiving the dominant or defective allele from the mother. Okay, that's it for the X-linked diseases. Thank you for listening.